You ladies and gentlemen are my first early viewers, so you get to see Edward this morning. He wanted to say hello. He's glad you're all here. He is very sorry to call a last minute audible, but he's also very happy that I'm working from home today. So I wish Edward would have. I told him he was sitting in my chair. He said, "Come on, Edward, you got to do some work. You're going to help teach the class." He doesn't have thumbs, so can't do much. We'll wait for sure until 8:30 until everybody gets on. Again, I'm recording this just so I have reference to go back. I'll post the video for you guys to look at later, but also so that I can go back and mark on my assignment sheet for attendance. So we'll give everybody a couple more minutes. I have one more announcement going out at 8:30, so please bear with me. I know it's a lot of emails, but I just wanted to make sure I caught everybody as best as I could. So we'll start at 8:30. If you're just joining us, please make sure to read the welcome note. Again, please, if possible, put your cameras on. This is a way that is engaging. I know it's nice to be at home, but again, we would have been in class and I would have been seeing you. So please make sure to just have your cameras on. And again, I am recording today's lecture so that we are able to have reference to it later, which is nice, but also so that I can give you guys points for attendance as well. So I am grateful all of you were able to, most importantly, I'm grateful for a couple of things. One, I'm grateful I was 5 minutes from home. I'm grateful that I had at least an hour to give you guys a heads up. Hopefully this saved some of you if others of you are at school or dorms. Again, I deeply apologize. This is really frustrating because I just bought these tires. I literally just bought these brand new tires to put on and had them put on 2 weeks ago. So such is life. But it does seem like I can save the tires. So I'm going to go to a patch shop quickly. I'm going to call Costco and see what their customer service is. Not that I'm sure you guys care all that much, but I was just like, gosh darn it, I just bought these tires. I was so excited, right? To adults and get new tires on my car. And then the universe had other plans for me today, so that's okay. We keep it moving. I'm grateful that I am safe. That is my number one, that I wasn't on the expressway and it blew out and I was in a precarious situation. So, and again, for those, you get to see Eddie. Eddie is happy you're here. He's not happy he's in class, but he's happy you guys are here. So with that being said, the in-class assignment we were going to do today, which would have been the case study for molecular biology, we will do on Wednesday, but we should have a good chunk to finish up chapter 3 today for molecular biology, and then we will start chapter 4 for tissues. We'll get pretty far through chapter 4 on Wednesday, and then on Monday you guys will come to lab. Hopefully we'll have no snow or anything of that nature and all things go well. And you guys will do a lab on DNA fingerprint analysis for the tissues lab. And then we will wrap up the rest of the material as we head towards spring break. And your next exam will be on the Wednesday prior to spring break, which is crazy. We are already almost halfway through the semester. So With that being said, I am going to go ahead and share my screen. Just give me a thumbs up if you guys can see it. Beautiful. Thank you. Okay, so we had started our conversation on molecular biology, and we were talking about DNA analysis and how we can use DNA analysis to say something about, of course, the genetic components within a crime scene. And again, this will lead perfectly into tissues and where we can obtain DNA, even though we've already touched a little bit about it when we talked about blood and the different areas inside of our different type of blood cells that we can obtain DNA. So we had left off, oops, this is the very end of the presentation, sorry. All right, we had moved through, just as a quick recap, we had moved through the PCR process. We had talked about how, again, we go through these several different steps of finding the different locations of the genome, finding the different regions that we want to amplify across, right? And that to do that, we have to have specific sequences that can copy and paste those gene regions, those loci, over and over and over again to become enough material so that we can visually see it with inside of a gel, right? And again, we talked a little bit about the PCR ingredients. So again, for our polymerase chain reaction, what do we need to have? First, you need to start with something. This is why collecting information at the crime scene is incredibly important because, again, what is the quality of the DNA? Has the DNA been degraded over time? Do we have a possibility that, we are looking at a cold case and that we don't have a complete sample? Do we have a mixed sample of tissue, right? Do we have the victim, the suspect? Are we in a public place? So again, what we are starting with is going to be very important. And so we need a DNA template. We need a physical sample to start with. Then what are we going to need? We're going to need something to unwind our DNA that we have so that we can make copies of both sides. And then what else do we need? Well, just like when we make a copy at the copy machine, right? We have a starting material of a book. That book has ink and has words inside of it. Well, now we need to copy that. So what do we need to make sure we have? We need to make sure we have paper that can make the copy. We need to make sure that we've got a scanner that can actually visually see what we are trying to copy. And we have to make sure that we have ink and the capability to print out those words, those letters in that order to make the exact replicate of what we see on the book itself. So think of your DNTPs, these deoxynucleotides, as again, just a bucket of those letters of A's and T's and C's and G's to pull from the copy machine to put down as we make a new copy of each side, right? So here's my original DNA sequence. I'm going to unwind it from some Taq polymerase through the denaturing phase. I'm going to add some primers that are going to say, hey, start here, right? And then I'm going to go to my bucket of DNTPs, and those are going to be the ink that are going to copy and make another new duplicate of each side of my DNA. So now my one becomes 2 copies, right? And now what do we see? From my 2 copies, I can open those up and make 4 copies, right? And again, we do this over and over and over and over again by binding the primers, binding those DNTPs together to make copies. And then we extend that out for multiple rounds until we eventually get nice big pieces of DNA so that we can visually see that. So again, question I can ask you from here. If I have a PCR and I'm running a PCR and I don't have my DNA template, can I start the reaction? No, right? If I have a PCR reaction, I have DNA template, Taq polymerase, DNTPs, primers and buffer in water, but I realized that my primer sequence is not right. Will the DNA PCR reaction run? Yes, the PCR reaction will still run, but you might not amplify the region that you want, right? So again, think through different ways you can strategize this information. If it was a multiple choice, if it was a true false, if it was a fill in the blank, if it was a matching, or if it was an essay question. Any questions here? And again, I'll try and monitor both chat and also please feel free to use the raise the hand feature if also there's any other questions. So as I had mentioned, I put in here for you guys a nice, very beautiful, simplified Amoeba Sisters. They break things down very straightforward. So if this is still a little bit confusing, please watch this. They do a fantastic job of explaining that process. Okay, we had then mentioned we need to visually see this. We had talked about how our DNA needs to be loaded into a gel. And again, questions I can ask you from this information, right? What is the arrangement? How do we need to set up the gel? So maybe I ask you, I have a gel box. You want to run a PCR. And you put the gel into the gel box. You load up your samples, right? And now what do you see? You start the machine, right? But you accidentally switch the positive and the negative such that the positive is up here and the negative is down here. What do we think is going to happen? What do we think the outcome would be if everything worked? I did my PCR correction, my reaction correctly. I ran it with all the things. I've loaded it into the different wells. And now I put it into this machine, but I accidentally have flipped. This is positive here and this is negative here. What do we think the output would be? You can put it in the chat. You can raise your hand. What do we think is going to happen with our PCR reaction if I flip my positive and my negative? Right? Let's see. No movement. Yes, you are correct, right? So what we would see, and this has happened, I have absolutely done this as a bonehead, right? If you join the lab and if you are in a lab for the first time and you're doing this for the first time, you bet your biscuits. Everyone can attest to the fact that they have accidentally put the cover on upside down with the positive at the top and the negative at the bottom. And what do we see? All of the DNA sample just stays at the top. It doesn't move because it needs to run like a battery from negative to positive in order to pull that DNA through the gel. So again, that would be a great style version of question of, again, what could go wrong if I don't do this correctly, right? This could be a human error that could lead to misrepresentation, right? Be sure to review, and I've got some questions at the end, again, of how to read, the different size, right? Maybe I put this as an image on the exam and I ask you to, give me the different sizes based on a sample. Again, looking at this, we can see that this one has 2 bands that are hovering that 60 base pair mark. So again, these 2 base pair bands would be maybe between, I don't know, 62, 63 for the upper band and maybe 58, 59 for that lower band, right? Versus DNA sample number 2, we've got 2 different bands that are very far apart. And again, if I asked you this, I would give you enough information to be able to say which band you were looking at, which sample you were looking at. Are you looking at the latter, right? And what is this particular gel image missing? What do we need probably in lane number 4 over here? What do we say is very important to make sure to run alongside of our samples? A control, right? And our control would be water, right? So we would still have all the other parts of the PCR. We'd have the DNA template. We'd have Nope, I'm sorry, take that back. We would have everything but the DNA template, and we would be putting in water in place of the DNA template to say that again, we see no bands because we didn't load anything inside of there. The last thing to point out here, right, is we're looking at 1/2, right? So we're looking when we run, we're looking at our A's and U's and C's, G's. Okay, the very last little bits that we ran through, I'm not going to take too much time here, is just how then do we identify STRs and SNPs. We said that our STRs, again, are stretches of DNA that are repetitive, and they are repeats that are next to each other. That's that tandem word that we're looking for. We also said that we are looking for variability because, again, things that are people are unrelated, there is going to be lots of degrees of variability with inside of these regions, giving us a very significant version of PM that tells us we are looking at two individuals, right? Whether it's uniquely victim or suspect. STRs occur on all of the 23 chromosomes. 22 autosomes are sex chromosomes as well. Again, make sure to move through this information because, again, as we said, there is some variability that we can see, but we do turn to ameliogenin as the gene region on our sex determining regions found within inside of teeth, or we can turn to the ameliogenin gene to be able to identify sex, but that we have to be careful because there are certain situations, certain populations that may have variations with inside of this. So again, everything we talk about, just a first pass, so to speak, of supporting evidence. Examples, we moved through some examples. We looked at some pros and cons. We looked at why STRs and how we can compare this in cases of paternity. And the last thing we were talking about was our single nucleotide polymorphisms. And again, the major difference here being that the terminology, use the full description, right? Single, one base pair, nucleotide polymorphism. So again, the difference here is we're not looking at tandem repeats. We're looking at 1A, 1T, 1C, or 1G. Finally, we had ended on the mitochondrial DNA, how this is a unique comparative analysis to our nuclear DNA. And again, questions I would ask you here, how much, compare and contrast the amount of mitochondrial DNA versus nuclear DNA. We had said our mitochondrial DNA is only about 16,500 base pairs, a lot smaller compared to our nuclear DNA. And for our mitochondrial DNA, we are only going to one specific location. Instead of the multiple locations inside of the nuclear DNA, we are only going to one place, and that is the D loop with inside of the mitochondrial circular DNA. Questions here? So again, maybe it's a compare and contrast. Maybe it's, how many different regions between the two. Maybe it's tell me the difference in base pair number, right? All the different kind of ways you can think about those questions. Maybe you just want to make a table, nuclear DNA, mitochondrial DNA, and just throw as much as you can on the wall, right? The very last thing we talked about was the fact that most of the mitochondrial DNA, if not all, is exclusively from the mother, right? But we do have instances where, in fact, we can see representation of individuals, mothers, who have healthy nuclear DNA, but they may have a disease inside of recombination that they give a higher prevalence rate of potentially giving an egg that has a mitochondrial disease. If they remove that egg, they can actually take out the mitochondrial DNA, still have the nuclear DNA of the mom, and they can actually put in a healthy patient's mitochondria, not the nuclear, and we can see that we can have an outcome of a patient or an individual, an offspring that can still be sperm and egg coming together from mom and dad, a nuclear 2323. But again, because mitochondrial DNA, from what we know, from what we understand, is that again, it is an ancient bacterial DNA that has been engulfed inside of us, and it is uniquely separate. How do we know that? Because again, it has this arrangement. It only has one location that we can go to. It's much smaller. It's its own unique system, right? Which allows for this to occur. So again, questions I could ask you here, maybe explain donor mitochondrial DNA. How does it work? What is being replaced? It is just the mitochondrial DNA from the mother with the mitochondrial DNA from a donor. There is no nuclear DNA coming from the donor. Why? Why would that not make sense? Why would I only be looking at this, mitochondrial versus nuclear? What happens in that situation? What would I end up with if I transferred nuclear DNA into an egg of a donor that's already got nuclear DNA? Does that equal viable? Would that survive? No, it's way too much genetic information, right? We would have too much genetic information. The egg would not survive, right? So again, just understanding that. And again, what could be challenges here? If I am turning to something, say, what's the type of blood that only has mitochondrial DNA? Do we remember from our last exam? Well, let's start with that box again. Do we have DNA, nuclear, and mitochondrial or mitochondrial inside of our red blood cells? No, right? Do we have nuclear DNA or mitochondrial DNA in our white blood cells? Right? Yes or no in there? And then how does that compare to our thrombocytes, right? So if we were looking at the differences between those blood cells, right? If we were looking at how our platelets, right, with just our mitochondrial DNA compared to our leukocytes, right, that need both because they are our defenders, they are our fighters, our white blood cells, right? If we only for some reason came across platelets at a scene, which would be pretty unique, I'll be very honest with you, right? And we were observing those and we were trying to obtain DNA from them, and we just happened to be across an individual who had donor mitochondrial DNA, there could be some confusion, right? We could be confused about potential identity, etc. So this is a very extreme case, but it is something that we have to potentially consider. All right, what other forensic insight can we gain? We had left off this, I think this was the very last one we left off with, right? We can say something about, as far as molecular DNA, as far as our genomics, right? As far as who we are, what we are, we can potentially look at ethnicity. We can definitely look at pigmentation potentially. We can look at underlying characteristics. We could look at disease states, right? What I'm showing you in this picture is, though, right, is what we have is known as the epigenetic component. So you have your DNA, the code is the code is the code. Then what do we have? We have all of these other things that can affect how your genes are expressed, right? So as we're opening up the DNA and printing out eye color, maybe again, all of these different factors can vary how it affects that gene getting processed and made. So what does that mean? It just means that really trying to come up with the physical appearance of an individual based only on DNA is really challenging because, again, look at all these things that could affect the way the genome is read. So we have to be pretty careful. Now, again, pigmentation or potentially eye color. Again, if we have a full nuclear scope, sure, we can gravitate getting some general understanding of those things. Again, it's challenging because as we had said in last class, pigmentation even across our own skin can be varied, right? So again, very challenging and not a first pass where we would go. Another really fascinating area that people are interested in kind of the overall aspects of genomics is psychopathy and what's the other one? Why am I drawing it? Psychopathic and sociopathic. Sorry, my brain took a complete dive right there. We're always fascinated of trying to understand, right? What is the underlying brain mechanisms of individuals? How can we use behavior? How can we understand genetic influences of the mind that output behavior, output cognition? And can we see differences? This is a very unique field of cognitive behavioral psychology. There is some movement in it, but again, what do we need in order to be able to say some of this stuff? We need a human brain. We need the person's physical brain. And again, there are plenty of people who have probably characteristics of being sociopathic and psychopathic walking around, but we're not getting access to their brains after death, right? Not always is the output going to be serial killer, of course. And so we have to be very careful, but there are some things that have led to potentially seeing predisposition, or maybe that enough characterization between behavior, environment, and overall genetics, we can see that there is some foundational understanding of these things. What do I mean? Again, they can influence certain things, but there's no single factor that is going to guarantee if you have this, then it's going to equal that, right? Correlation does not equal causation. There's plenty of instances where this occurs, but we don't get the output of serial killer. Now, let's look at some examples. So in some men with genetic variation linked to a low MAOA, monoamine oxidase, right? So again, this is something that helps to break down your monoamines inside of your brain, things like dopamine, things like serotonin, right? This is something that helps to reuptake. So if there is some variability within that specific enzyme, and there are individuals who have that variability, who experience childhood mistreatment, if those two things compound together, what we have found is there is some research that shows in men specifically that, again, there is a potential of an increased risk of violent behavior in adults. I'm not saying it guaranteed, but there has been enough genetic linkage or enough studying of saying, okay, what was background of individual? Let's look at all these different genes that express inside of the brain for all these different neurotransmitters and how these neurotransmitters work. Let's see how different brain regions light up under certain functionality, right? And again, notice there's lots of big if statements in there. Some, may, right? Not absolute. There has also been research that has looked at men with variation with inside of the SERT gene. So again, this is another transporter that takes up excess when serotonin is released in your brain. It gets reuptaken back into, and sometimes how much we can load serotonin to be released, right? Maybe we're not putting enough into the vessel vesicle before it releases to allow for serotonin. Again, serotonin is the love drug. It makes you feel good. It reduces stress output, right? It allows for emotional connection. These things are very important that we see lacking in a lot of individuals who have sociopathy and psychopathy, right? So again, if they have variation, not always, there's a big slide scale of that variation, coupled with stressful childhood events, may have a higher likelihood of suicide and depression. So again, we can't be 100% certain, because are there men with variation in the SERT gene that have stressful childhood events that are depressive and suicidal? No, right? There are plenty of men that exist without that. But these are potential big buckets we can start to look at, is all I'm saying. So again, these correlations do not mean outcome, right? Genetics alone does not determine your behavior or mental health. This goes back to epigenetics. So I want to make sure to point out here. I remember in the very first time that we were talking about the nuclear DNA, we had said 16. For the purposes of CODIS, which is the online system that looks for gene regions. Even though there are potentially 16 different regions, again, we are missing 1, we're missing 9, we're missing 10, we're missing 14 that were represented in that first picture. It is in fact 13 regions that the CODIS system is going to look to identify. So again, the Combined DNA Index System, this is a system that is a law enforcement system that is specifically now taking those 16 known variable regions and actually bringing it down to 13. So it's even reducing that number even further, because these are the most varied regions that they understand and know to give, again, that correct PM value. So again, if I'm going to ask you the question on the exam, even though there are 16 known varied regions, the CODIS system is only using 13. We still have ameliogenin. Again, ameliogenin is still on that X and Y that is going to be for gender. And again, why are these chosen? Because these are all highly varied regions. So there are about 12.5 million profiles that have helped in over 224,000. I'm sure that number is much higher now. And again, I put this in here for you, but this is just kind of a thought of, well, what is the ethical implication here? We had mentioned that when you do 23andMe, in the long list of, you have to sign off and say, like when you get your contract with Apple, right? Your agreement. I don't know how many people are really reading through all that, but if you read through your 23andMe, there is language in there that basically says you're an open resource now. Your DNA is now, can be put in, can be utilized, right? And so this was, again, the conversation we had about cold case files. The individuals sometimes get the knock on the door and they're like, hey, who's uncle or who's aunt so-and-so? How are they related to you? When's the last time you saw them? You did this 23andMe, and it turns out you're a pretty solid match to this cold case we have in our system. Has happened, right? But again, what's the ethical implications of those things? Familial searching, right? So again, family members share the same genetic profile. And if we are going to look at this, this is a situation by which maybe we find that a person is in the system, but they aren't the person that committed the crime. If a person that is already in the system that is a family member that is linking, right? So now we're not talking about 23andMe, we're just talking about the CODIS system. This is really important because, again, we are looking at, compared with all other records, we're looking at a different subset of profile. We're looking at familial searches. That would be the search bar indicator that we would, look at things that align up to, say, close relation, right? This is also based on 3 major assumptions: that if we find a person that is in the system, we potentially suspect that a family member is a potential suspect here, right? And we see a hit coming up, but their alibi, or maybe they're physically already in the prison system, so there is no way they could have, caused this crime or committed this crime. Now we have to go out, right? They're coming up in the search, so it's like, okay, what's going on? There must be a family member that's committing this crime. And this again is based on potential assumptions. Most crimes tend to be committed by somebody from a local area within the same proximity, within a, family lives tend to live close together. Criminal behavior, unfortunately, will sometimes often run in families, right? If it's generational, this is another thing that is just an assumption that, investigators work on. And most recorded crimes are by or on individuals with low SES. What is SES? Do we know what SES is? Have we ever heard the term SES before? Anybody have an idea? SES. It stands for socioeconomic status. So again, most crimes are by and on individuals with lower socioeconomic status. What does that mean? Individuals who cannot vocalize and advocate for themselves. So individuals that maybe come from poor communities, violent communities, neglected communities, right? And unfortunately, individuals, not always, but again, assumptions here. Individuals that are of low SES status tend to stay within low SES status. It's not always, again, correlation is not causation. These are assumptions. So these are just questions that, for food for thought. I don't know. Are we moving towards this? Do we think these are valid points? Let's just answer the first one. Put in yes or no to me inside the chat. Should all citizens be required to submit DNA samples for national databases? I see a lot of no's right out the bat. Okay, can anybody give me a why? Why would you be adamant about no? Because you guys very much know, which I agree with. But what is your justification for that? Invasion of privacy? Anything else? Great points, guys. Excellent points. Absolutely. What if, we definitely have seen situations by which sometimes those in the justice system are not necessarily the most truthful or most honest, right? Or again, if you are an individual that knows you can get access to a database or access to the storage facility, yeah, absolutely. What if I can take your DNA, right? We've seen enough, even James Bond, what's the other guy? I can't think. Tom Cruise always plays him. I don't know. I can't remember his name. But, they come up with those, you can pull and lift off prints of other people and use those prints to open. Yeah, absolutely. Right? Is there already potential exposure to things like this that we're experiencing? And if so, where? You may not have, necessarily submitted a direct sample of your blood, right? Hospitals, yes, potentially. Thank God for right now, we still have, HIPAA that protects, medical and patient doctor privilege and all of that good stuff, which thank God that still upholds in a court of law. Where else are we kind of freely giving access of our information to? And now think about it. Think about how many of your apps, right? At first, it was just what? The phone that needed this to open, right? It was just Apple that said, hey, let's put this safety measure in. Now, what do you see? Every single application says, do you want to just turn on Face ID? Make it easier for you to come in? So now guess what? Every one of those apps has access to your retinal scan. Your retinal scan is just like a DNA fingerprint. It is very unique to you. So again, they've got access to this stuff. I don't know. Is that another thing we need to raise, right? So what would I ask you about this? Maybe I have just, this is an extra credit question. Maybe it's a food for thought type of question and you have to kind of walk me through it. So again, when you're coming to study this information, think through, right? Try and justify this. The more information you give me as an exam question, the better chance I have to find details to give you the answer, or to give you the credit for the answer, I should say. Okay, so let's get back to Earl, right, to wrap up our case for this particular chapter. So for Earl's case, right, there were many people that advocated for him. Again, even back in the 80s when DNA analysis wasn't necessarily at the forefront of presentation as evidence, there were still people that were definitely fighting and advocating. I mean, of course, his family, but there were people that were fighting and advocating for him to say, clearly this gentleman did not do this, right? And so remember, he was potentially going to go, he was put onto death row. He was in the queue, so to speak, for death row. So forensic methodology that they used once it became more readily available. They used DNA extraction. Obviously, they used the PCR, STR, and then they also used this random match probability, which we have talked about, right? And so this is the overall kind of timeline of STR analysis, which brings us up to how they used it in Earl's case. So even though we can see that there's some kind of jumping around of advancements for Earl, we see that really we didn't get till 1993 until we started using this DNA analysis. Remember, we were all the way back in the early 80s for him when we had put him potentially behind bars. He had given sort of the false testimony. He couldn't line up the crime scene. He couldn't line up the victim. He couldn't line up that her children were there. So really it was a lot of advocacy on behalf of him. And it wasn't until 2000 that they actually were able to prove that he was innocent. This led to his ruling and his dismissal of the case in 2002. The Supreme Court ruled to put bans on the death penalty for individuals with intellectual disability. Now, does that solve the problem that we're even having individual with disabilities? Not to say that there aren't those that commit crimes, but that we are not addressing the fact that they could be giving false testimony or they could be admitting to things that they don't understand, right? It's still not addressing those things, right? But it's simply just saying, maybe if we got it wrong, we can't put them on death row. Take that however you want. I don't know if that solves the problem, but there is at least law in order now. He was released on February 12th in 2001. He did get married. I don't know if this is still the case, but last reported he lived in Virginia Beach, and he was awarded through, of course, wrongful suit, $2.25 million in settlement against in 2001. So I don't know what that would equate to in today's dollars, but in that particular time, that's what he was awarded. So we will pick up on Wednesday and do the group handout. So with this being said, now we're going to transition over, and what you'll see at the remaining part of this, I've got some practice questions, I've got some videos, I've got some kind of repeat information, right? Looking at things. This is all potentially great, practice for getting prepared for the exam. Any questions on chapter 2, or I'm sorry, chapter 3? Okay. So with that, then we're going to start and get into human tissues, and this will be, again, the foundation for a lot of the things we're going to look at, especially when it comes to DNA fingerprint analysis for our lab on Monday. So the biggest things that we're going to take away, we're going to look at our 4 major primary tissues that we're going to look at as far as evidence, look at the applications for forensic analysis, and identify some key characteristics that would help us to tell us something about the investigation. So just a high level overview, because again, where are we? We are in biology. That is the foundation for this course. So just to recap back on our anatomy, physiology, a little bit of biology, right? If we are looking at what we use to define the terminology of tissue, tissues are a group of specialized cells that perform a collective function, right? So we have different tissues. Your blood is a tissue because it is very unique to just doing its job as blood, right? Again, you are a eukaryotic organism. You are multicellular. What does that mean? You have lots of different cell types that come together to make your body, your organism function. And what does that mean? You have brain cells, you have stomach cells, you have heart cells, you have different types of cells, multicellular, that come together as one organism, right? You are not a bacterium, which is one entire organism as one cell. Again, just to kind of remember where we are. And again, these are crucial because these are areas that we can go to provide information about the crime scene. So in looking at our kind of high level overview of our 4 different types of tissues, right, that we're going to kind of look towards for the purposes of this course, we're going to look at things like our epithelial tissue. We're going to look at, very singular cells that come together to create sheets that cover and create surfaces. We also have connective tissue that, of course, use the word to your advantage. It binds and supports and provides stability. Muscle tissue provides your movement. We have, again, things that are under voluntary and things that are under involuntary control. I'm sure you're all grateful that you don't have to think about doing digestion. And then we have nervous tissue. We have things that innervate into these other tissues that provide feedback to our brain to tell us what's going on in our environment, both externally and internally. So epithelial tissue, when we look at our epithelial tissue, again, it is coating surfaces. It provides skin, right? It's the outermost portion. What is the function of what the overall explanation of epithelial tissue? And again, epithelial tissue is not just skin, it can coat surfaces, right? It provides protection. It can do secretion. It can provide absorption. It can filter. Things like our skin, of course, the biggest one. Glands are coated in this, lining of organ systems, right? Usually the foundation of a lot of where things come together to adhere is going to be epithelial tissue. Very single, thin layers to help support and again provide sort of structural coverings to do protection, secretion, absorption, and filtration. So why is this important? Again, we can use this to, we can use these locations specifically, right? We usually tend to think about skin for identify things like burns, bruises, chemical exposures, right? What was your body exposed to potentially? And it also can tell us things like disease states. So for the JonBenét Ramsey case, if we weren't familiar or we haven't remembered this, this was a very famous case back in 1996, still unsolved to this day. There was DNA underneath her fingernails and in her underwear. Still unidentified as to who. Which is, it's still very interesting that this case is open. Lots of very interesting timeline around this story. But, even as much as we would, we look to, if you've ever watched any sort of documentation on it, or you've ever watched a documentary, like, again, the family all acted in odd behavior, but none of the DNA from them matches this. It's a very interesting, odd case in a very, very small town in Golden, Colorado. Still unsolved. Still to this day. So what do we know about the skin? The skin has two major layers. It has the epidermal layer, the outermost, and the dermal layer, the innermost layer. So the epidermis is the one that's constantly shedding, right? This is the thing that's giving dust particles in your house. This is the dander. This is the same thing from your pets. If you have pets in your house, right? All that dust is typically dead skin cells, right? Things that have been shed off because your epidermal layer is replenished, right? It pushes out the old to replenish the new. How do you know that's true? Have you ever gotten a cut, right? And watching it heal, it eventually seals back up. The dermal layer is going to be down here where we see, again, a lot of more of the deeper stuff like our blood vasculature, our fat. We're going to be adhering to the base layer of muscle. And again, where can we pull out of? What can we look to in the skin? Because again, what does our epithelial tissue do? It protects, it secretes, it absorbs, and it filters. So sweat, right? Sweat is a big one. Sweat comes out of our skin. It helps us to thermoregulate. And it is important because, again, we can look at the balance of urea, salts, and water. We can look at the content of that. The skin is by far and away the largest organ of the body. It is an organ. It is something that serves a very specific and unique function compared to all the other organs in your body. And as mentioned, protection, temperature regulation, sensory reception. Forensics, how do we use this? Because again, the skin is a map. It can tell us what the body has been through. If again, we are still in the stages where we can obtain the body to be in a place where we can look at the skin, right? We'll definitely, if we're in the later stages of decom, we're not going to be able to identify these things. So as mentioned, again, making sure that we know the difference, things I could test you on, right? In our epidermal, our upper layer, right? We are looking at things that are keratinized, stratified epithelium. What does stratified mean? It means, look at this picture. They are just stacked on top of each other. They're single cells that are stacked like bricks, stratified, right? They're not just one singular layer. It's one cell, but it's kind of stacked like a brick on top of another cell. Stratification, right? Layers. Inside of our epidermis, what do we have? We have melanocytes, those that give us our skin color, pigmentation. And we have keratinocytes, sorry. So again, things that provide keratin, cuticle, things that give more of that robust nature of skin. It's not as delicate, right? You can push and move skin. It's got a little bit of resistance, residual resistance to it. And keratin is good because it helps to keep the moisture in the body versus drifting away, right? So we want a little bit of that almost layer with inside of there that helps to keep that sort of waxy kind of binding It's not wax, but it's necessary. It's keratin, and it keeps the moisture inside. As we get down to, again, the dermal layer, notice here, avascular. I do not have vascularization. I do not have blood flow in my epidermis. I have blood flow in my dermal layer. And again, making new cells, fibroblasts, fat cells, fighters, my macrophages, things that are going to gobble up things that shouldn't be there. So again, all of the epidermis is a big, thick layer of skin that's constantly, right? Our cells are constantly moving up because they're being replenished from the dermal layer to make sure that this coating is always protected. I want to pause here. Any questions? So what does the epidermis tell us? How is this important? Again, as we move to that outer cell layer, right? That dead skin, and especially again, if we see it in sort of individuals who have built up calluses on their hands, right? By roughing that surface up over and over and over. Well, you maybe aren't able to replace it fast enough. So what does the body do? The body creates a very rough callus to keep your skin protected. But what is this dead skin made of? What is this dead skin building up? It's building up into a cuticle, rough, right? Especially even on your hands, even if you have very, very, very soft hands, you might feel along the ridges of your hands, you might feel a little bit of raised because you use this part of your hand a lot, right? If you don't feel it on your hands, you probably for sure would feel it on your feet. Your feet have a nice big thick cuticle because you are walking around, right? You're barefoot. You want to keep your feet protected. Evolution has said, put a nice big thick cuticle there because we didn't have shoes for a very long time of our evolutionary history. The thickness of your skin is going to vary across the body, right? Where are places that we imagine we have the thinnest skin? Where do we have the thinnest skin represented on the body? Eyelids for sure. Eye cavities, right? Where else? Pretty much the face in general.
Does the top of our hands count?
Absolutely, right? Think about elderly individuals. This is one of the first places that we tend to see bruising, breakage, right? This one's very, very protective and very dangerous. Skin against the shins, skins on top of our feet. And then again, most importantly, genital areas, right? Those are very sensitive, very, very thin skin. So again, we can look to these different regions to look for, again, distribution of patterns, exposure, etc. Repetitive abrasions, we can also use these as indicators. What did the person do in their life? So if we look at this picture here, what does this kind of look like? If you saw these indentation marks on a body, right?
Guitar?
Yeah, somebody that plays some version of the strings. And in fact, it is the hand of this individual. What do we think this individual does? Yeah, she's holding it at the 3 points to play the violin. But again, if we didn't know and we weren't taking in these observations, if I didn't look at this, I could potentially mischaracterize what those are, right? I could say they look like some version of strangulation. They look like something nefarious has happened. Oh, take the rest of the observation. Oh no, she's got indentations. I wonder if she played some sort of instrument. Again, additionally, these things can tell us what was the lifestyle of an individual, because then that ties us back to who can I pull to and talk to about timeline, right? PMI is always our end goal. The unfortunate part is, yes, unfortunately, we can see scarring as part of torture, right? What do we think these marks are?
Cigarette butts.
Cigarette burns, maybe some sort of, I mean, yes, they are cigarette burns just based on the characterization of the darker versus the outer light, right? But sometimes individuals, may drop nefarious, types of liquids on individuals. There's all sorts, unfortunately, that can tell us about lifestyle choices. We also can look to the skin to tell us something about an individual by what they identified as or who they identified with, right? Tattoos tell us a lot. Usually people choose tattoos on their body because they have significant meaning to them, right? So again, they can tell us about their race. They can tell us about their religion, nationality, their occupation. Sometimes people even put their own names on their body. Sometimes they put the loved ones of individuals that they have, right? Sometimes they'll put dates on their body, right? Again, your body, your choice to do whatever you'd like with it. But these are all very important for building the story of who this person was in life. It can also tell us a little bit about the mental capacity of a person, meaning, did they go through experiences? So again, the semicolon, does anybody know what the semicolon represents?
Suicide prevention.
It is actually something for a contemplation of suicide, right? So the semicolon means a continuation of a sentence, right? And so the semicolon in the case of a mental status is sometimes what people will put and choose as a tattoo to state that, they're contemplated or a survivor of attempted suicide. Again, look at some of these other things, right? We can even have individuals who have gone through a traumatic experience where, again, something can happen and they can create such an injury that it can impact and leave a physical tattoo, right? If you've ever seen images of individuals who have ever been struck by lightning, they tend to get very bizarre patterns of where the electricity physically went through and sizzled, burned their vasculature, and you can see it forevermore, right? Again, not only this, we can look at individuals that maybe are potentially using tattoos to hide drug addiction, sexual preferences. And again, we can tie back to even potentially where you got your tattoo from, right? Who was the shop? Who is the individual? Because again, these are all potentially people we need to speak to about who you were, where you were, what you were doing, right? Of course, most notoriously, we can look at gang affiliation. So individuals that have, marked themselves in an identification or alliance with a particular gang. Very important, especially when it comes to either suspects or victims. Sometimes we see these before and or after prison. Sometimes individuals will get these tattoo markings while in prison as well. Maybe they get out and reoffend. So again, we can look and identify these things, and we can also look at things like our hate symbols. Because again, they all tell us something about what you align with. Whether you are an individual that is the victim that's got these on your body, or we are looking for a potential suspect, right? Maybe you are a person that was the victim of a hate crime and you were branded with one of these, right? Sometimes we see that as well. Or sometimes we see inside of rooms, there's branding of particular hate, symbols put into particular rooms, right? To note, feelings, justification, or etc. towards victim and suspect. Lastly, we can also look at scars. Scars tell us a lot. Scars are very much an identifying marker to us as far as telling us about an individual, right? Up here, this tells us a lot about mental status, right? What is this a picture of? Self-harm, absolutely, right? This is an individual who is known as a cutter. They are taking a razor blade and making tiny cuts on themselves to inflict pain, also to provide relief, right? So again, self-torture is a part of something that we have to identify as part of mental status. Um, most most cutters will go to very thin tissue, but also some people don't want this to be seen. So you'll have to look at other places on the body, right? They may hide this. Usually, again, individuals like this would be known for wearing very long sleeves. They don't want people to see this as an identifier of them. But again, scars can tell us a lot. They can help us to identify a body. We don't have much, and we are looking for people to identify our victim, right? Loved ones, people who are close to you, may know these markings on you and can identify you. It is very important. So forensic relevance of, again, our scars, they can indicate unique biology. Sometimes we just have scars on our body naturally from birth, from output, etc., right? And again, can indicate past injuries, can tell us about abuse. Absolutely important. And again, when we get to a certain component area of scars, depending on how deep, often what do we see? Depending on how far down we're going into our epidermal and dermal layers, if it's inside the epidermis, we're going to replenish that tissue. We're going to replace that skin, thankfully. But if we go down far enough into the dermal layers, we're going to see that we might not have hair follicles anymore. We might not have our sweat glands, making them distinguishable from normal skin. So again, if you have scars on your body that were unfortunately deep enough, you can look at them and say, oh, it feels a little funky, right? It doesn't heal the right way anymore. And maybe it doesn't have hair growing out of it the way that all the rest of your skin around it does. It has different color formation because, again, it had to come from a very deep layer. You bypassed all the layers that normally would have been replenished. Okay, so that leads us into, and again, the next couple of slides that will take us home for Wednesday and will give us a great foundation for our lab on Monday. Now let's get to what we probably notoriously identify or align with DNA or tissue analysis, which is our forensic fingerprint. Again, yes, your fingerprint is unique to you. There are no two fingerprints that are the same, but there are some characteristics that may be the same to other individuals. How are your fingerprints formed? Have you ever thought about that? How are your fingerprints actually formed? Well, when you are in utero, when you're growing as a baby, right? What's actually happening is that your skin on your fingertips is growing at a very fast rate. It's actually growing almost too quickly compared to the rest of the skin, right? And actually, as it does, the fluid and all of those other things that are grinding up against inside of the uterus, right, or inside of the embryonic sac, it actually is creating little waves because the skin is growing so fast, it's actually growing and folding over and growing and folding over. Now, that's a good thing because, again, you want a cuticle. You want it to be a little bit more thicker here because you're going to use this a lot. But that's how your fingerprints form. They actually form by the friction and the fast upward movement of this skin coming up in such fast in comparison to the rest of the skin around it, right? And compared to things on the back of your hand, that it actually creates these folds. And so we can absolutely identify and use fingerprints for personal identification. So I put this in here. You don't have to watch it, but I thought it was cool. It just physically does explain how fingerprints are created in utero. Something fun about it. Okay, we'll end with this today. So, fingerprinting, it is the foundation for basic acceptance. It's been around for a long time, right? This has been something, and again, when we get into lab on Monday, because there are so many easy ways to leave prints behind, to pick up prints, to identify prints through lots of very, very simple and straightforward methods. This has been something we've been turning to in a court of law for a very long time. The probability that two fingerprints could match is extremely low. It's estimated that there are about 64 billion different prints on planet Earth. Does that mean completely unique, right? Again, look at the ratio. How many people do we have on planet Earth? Right? That's why we're saying a very low probability here. We don't have 60. If we had 64 million people on planet Earth, we'd have a problem, guys. We'd be living on top of each other. It's way too many people. But again, the idea being that there is variation. So in the past 90 years in the FBI Central System, we have not found two identical prints. But, right, just leave you with this. We're going to pick up with this video next time, that sometimes there are things that are not perfectly unique. We have found sometimes that there is similarity. And we'll pick up with this on Wednesday. We'll finish the remaining portion of this chapter, and we'll also move through our case study that I had for you guys in class. So again, on Wednesday, the little case study you guys will be moving through will also serve as maybe a hint, hint, nudge, nudge, wink, wink, good indication to come. Foundation for some extra points on your next exam. So make sure you show up. Again, this has all been recorded, so I will post this as a video for you guys to go back to and watch again. Thank you for coming. Thank you for being amenable to my last minute nail in tire. I'm going to go deal with that now so that I can see you guys on Wednesday. But I will see you all on Wednesday. If you have any questions, please let me know. I'll hang out for a couple minutes if you have any additional questions for class. If not, have a great day. I will see you. Chapter 4 is posted. It's open, I believe. Have a good day, guys. Yeah, chapter 4 is posted. That's it. Let's see. Nope, I thought I did post it. Just kidding. Publish. I want to publish. Okay. Princess, are you there? Princess, again, you were supposed to have your video on and present. I might have to mark you absent for this since it's very clear you're not near your video. Okay, I'm ending the call.